Hi friends, this is Preeti. Today I am going to discuss about cloning vectors. Cloning vectors are the tools used in recombinant DNA technology. So the definition of cloning. Cloning is the process of producing copies of cells or DNA fragments. It is a multiplication. Then vector. The DNA used as a carrier for transforming fragment of foreign DNA into a suitable host is called as a vector. Vectors used for multiplying the foreign DNA sequences are called as cloning vectors. Here foreign DNA is nothing but the target DNA which we isolate from the human cell. Next, types of cloning vectors. Plasmid. It is a small, circular, self-replicating, extra chromosomal, double-stranded DNA molecule which carry small fragments of foreign DNA. So next cosmid. Cosmid is also a plasmid but which have a extra feature which is a cos sequence. It also carries small DNA fragments. Next bacteriophage. It is a virus which is used in the transduction process to transfer the genetic material from one bacteria to another bacteria. So it is used to carry medium DNA fragments and bacterial artificial chromosome and yeast artificial chromosome. These are used to carry large DNA fragments. Features that are required to facilitate the cloning in vector are origin of replication, selectable marker, multiple cloning site and molecular weight. So this is the structure of a basic plasmid. Here we can see the region for origin of replication. Here multiple cloning site where the restriction endonuclease can act, and this is the place where the target DNA is added. And this is the antibiotic resistance gene which is a selectable marker. First feature, origin of replication. This is the sequence from where the replication starts. This sequence is responsible for self-replication independent of chromosomal DNA. If any target DNA when linked up to this sequence can be made to replicate within the host cell. Many copies of target DNA can be recovered due to this feature of vector. Next is selectable marker. It is the region which contain antibiotic resistance gene. Plasmids containing antibiotic resistance gene are used in recombinant DNA technology. Selectable marker helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. Here transformants are nothing but the bacteria which took up the recombinant DNA. Nothing but plasmid containing the target DNA. For example, normally the E. coli cells will have the resistance against the antibiotic, but they don't have resistance against ampicillin, chloramphenicol, tetracycline and kanamycin. So when these cells are added to the media containing these antibiotics, they die. It means they are non-transformed and the transformed cells will survive. So the transformed cells means the plasmid having the resistance against these antibiotics will survive. The bacteria which took up the plasmids having these resistance against these antibiotics will survive. So it is the use of presence of selectable marker. Next, multiple cloning sites. It is the recognition site to link up the target DNA. These are the sites where different restriction endonuclease can act. Next, molecular weight. The cloning vector should have low molecular weight. Then only it can take up the DNA, the target DNA. And this is the plasmid vector, origin of replication, and this is the place where the restriction endonuclease can act, and here we can insert the target DNA, and this is the promoter region. It starts making RNA here and finally protein, and this is the selectable marker where antibacterial resistance gene or antibiotic resistance gene is present, which is used to differentiate between transformants and non-transformants. In the name itself, we can understand by the name selectable marker, means it selects the transformant cells from the non-transformants. Non-transformants are the cells which do not took up the plasmid containing the foreign DNA, and transformants are the cells which took up the plasmids containing the target DNA, nothing but recombinant DNA. Thank you.